DNA modifying enzymes and significance. DNA modification refers to changes or alterations occurring in DNA molecules during replication, transcription, repair, genetic recombination, epigenetic regulations, etc. DNA modifying enzymes, the enzymes that affect changes in the DNA, chemical constitution, or topology, are generally referred to as DNA modifying enzymes. Broadly, they can be grouped into two categories based upon the nature of modification performed. Composition modifiers. These are the enzymes that change the chemical constitution and require net gain or loss of covalent bonds. Major composition modifiers include enzyme classes such as nucleases, methyltransferase, methylase and demethylases, phosphatases, kinase, polymerase, and ligases. Then topology modifiers. They change the topology of the DNA molecule and does not result in net gain or loss of a covalent bond. These enzymes are primarily involved in DNA replication. They are generally classified into two classes based upon the number of DNA strands cut by the enzyme in single reaction. Topoisomerase type 1, single strand cut and seal, and topoisomerase type 2, double strand cut and seal. Important DNA modifying enzymes are nucleases. This class of enzymes break the phosphodiester bonds of the DNA backbone. They produce single and double stranded breaks in the target molecule. In living organisms, they are essential machinery for many aspects of DNA repair. Defects in certain nucleases can cause genetic instability. These are two primary classes of nucleases. Exonuclease. They attack at the end of nucleic acid and usually remove just a single nucleotide or sometimes a short oligonucleotide. Any particular exonucleotide, either the 3' end or exonuclease, either the 3' end or 5' end, not both. Val 31. Exonuclease 3. Endonuclease. Enzymes belong to this class of nucleases degrade or makes cuts at internal site. A number of different specificity endonucleases exists. Some examples are S1 nuclease. They cleave only one single-stranded DNA, including single-stranded nicks in double-strand DS molecule. Restriction endonuclease. They function to defend the cell from invading viral bacteriophages. They mostly cleave DS DNA, but only at a limited number of sequence-specific sites. The subcategory of endonuclease recognizes and/or cleaves DNA at specific sequences. Recognition sequences are restriction sites as compared to no sequence specificity in case of other nucleases. The restriction enzymes are further categorized into four types, type 1 to 4, based upon their properties. Now applications. Restriction endonucleases are used in the process of molecular cloning. Exonuclease 3 is used for generating single-stranded templates. Deoxyribonuclease. A nuclease enzyme that can catalyze the hydrolytic cleavage of phosphodiester bonds in the DNA backbone are known as deoxyribonuclease. Based on the position of action, these enzymes are broadly classified as endodeoxyribonuclease, cleave DNA sequence internally, and exodeoxyribonuclease, cleave the terminal nucleotides. Unlike restriction enzymes, DNase does not have any specific recognition or restriction site and cleave DNA sequence at random locations. There is a wide variety of deoxyribonucleases known, which have different substrate specificities, chemical mechanisms, and biological functions. They are deoxyribonuclease 1, DNase 1, an endonuclease which cleaves double-stranded DNA or single-stranded DNA. The cleavage preferentially occurs adjacent to pyrimidine CRT residues. The major products are 5' phospho-related bi-, tri, and tetranucleotides. Deoxyribonuclease 1. It requires divalent ions Ca2 plus and Mn2 plus and Mg2 plus for its activity and create blunt ends for 1-2 overhang sequence. DNA cell is most widely used enzyme in cloning experiments to remove DNA contamination from mRNA preparation, to be used for cDNA library preparation, northern hybridization, RT-PCR, etc. The mode of action of DNA cell varies according to the divalent cation used. Some of the common applications of DNA is one are eliminating DNA contamination, plasmid from preparations of RNA, analyzing the DNA protein interactions via DNA proofreading, nicking DNA prior to radio labeling by nick translation, etc. Then deoxyribonuclease 2, DNase 2. This is a non-specific endonuclease with optimal activity at acidic pH 4.5 to 5.5 and conserved from human to C. elegans. It does not require any divalent cation for its activity. DNase 2 initially introduces multiple single-stranded nicks in DNA backbone and finally generates 3' phosphate groups by hydrolyzing phosphodiester linkage. This enzyme releases 3' phosphate groups by hydrolyzing phosphodiester linkage and creating nicks in the DNA backbone. DNase 2 acts by generating multiple single-stranded nicks, followed by production of acid-soluble nucleotides and oligonucleotides. The catalytic site of the enzyme contains 3 histidine residues which are essential for enzyme activity. Some of the common applications of DNase 2 are in DNA fragmentation as molecular weight marker in cell apoptosis assays, etc. Then ribonuclease. Nucleases that can catalyze hydrolysis of ribonucleases from either single-stranded or double-stranded RNA sequences are called ribonuclease RNAs. RNAs are classified into 2 types depending on position of cleavage: endoribonuclease, cleavage internal bond, and exoribonuclease, cleavage terminal bond. 2 common types of ribonucleases are ribonuclease A, RNase A. It is an endoribonuclease that cleaves specifically single-stranded RNA at the 3' end of pyrimidine residue. Optimal temperature for RNase A is 60 degree, activity range 15 to 70 degree, and optimal pH 7.6. Then ribonuclease H, RNase H. It is a non-specific ribonuclease that degrades RNA by hydrolytic mechanism from DNA-RNA duplex, resulting in single-stranded DNA. Enzyme-bound divalent metal ion is a cofactor here. The product formed is 5' phosphorylated ssDNA. During cDNA library preparation from RNA sample, RNase H enzyme is used to cleave RNA strand of DNA-RNA duplex. RNase is important for RNA maturation and processing. RNase A and RNase H play important role in initial defense mechanism against RNA viral infection. They can be used to remove RNA contamination from DNA samples. Then DNA polymerases. They are a group of enzymes that are used to make copies of DNA templates, essentially used in DNA replication mechanisms. These enzymes make new copies of DNA from existing templates and also function by repairing the synthesizing DNA to prevent mutations. DNA polymerase catalyzes the formation of the phosphodiester bond, which makes up the backbone of DNA molecule. Types of DNA polymerase 1. This is a type A, family A polymerase enzyme. Its main function is excision repair of DNA strands in 3' or 5' and 5' to 3' direction. It also helps with maturation of Okazaki fragments, which are short DNA strands that make up the lagging strand during DNA replication. Its role during DNA replication is the addition of nucleotides at the RNA primer as it moves along the 5' to 3' direction. So, proteolytic cleavage of DNA polymerase 1 resulted in a fragment without 5' to 3' exonuclease activity known as Klenov fragment. DNA polymerase 2. It belongs to type A or family A polymerase enzyme. Its major function is the 5' to 3' to 5' exonuclease activity and to also restart replication after replication stops due to DNA strand damage. It is found in the replication fork to help in directing the activities of other polymerases. Then DNA polymerase 3. This is the primary enzyme that is used in DNA replication belonging to the family C or type C. It is responsible for the synthesis of new strands by adding nucleotides to the 3' OH group of the primer. It has 3' to 5' exonuclease activity, hence it can also proofread the errors that may arise during DNA strand replication. Then Taq DNA polymerase. This is a thermostable DNA polymerase 1 that was isolated from thermophilic bacteria Thermus aquaticus. It is commonly used in PCR to amplify short strands of DNA. Due to its thermophilic nature, it can withstand protein denaturation that is required during PCR. So DNA ligase catalyzes the formation of phosphodiester bond between two deoxynucleotide sequence of two DNA strands. DNA ligase enzyme require a free hydroxyl group at the 3' end of the DNA chain and a phosphate group at the 5' end of the other and require energy in the process. The role of DNA ligase is to seal nicks in the backbone of double-strand DNA after lagging strand formation to join the Okazaki fragments. This joining process is essential for the normal synthesis of DNA and for repairing damaged DNA. It has been exploited by genetic engineers to join DNA chains to form recombinant DNA molecules. The most widely used DNA ligase is isolated from T4 bacteriophages. T4 DNA ligase needs ATP as a cofactor. The enzyme from E. coli uses cofactor NAD. Except this, the catalysis mechanism is somewhat similar for both the ligases. Discontinuity repair. Discontinuity DNA ligase joining two molecules. DNA ligase significance. DNA ligase enzyme is used by cells to join the Okazaki fragments during DNA replication process. Used in repair of discontinuity. Missing phosphodiester bond in one strand joining two molecules of dsDNA together. In molecular cloning, ligase enzyme has been routinely used to construct a recombinant DNA. Then methyl transferase or methylase. Methyl transferase or methylase catalyzes the transfer of methyl group or CH3 to its substrate. The process of transfer of methyl group to its substrate is called methylation. Methylation is a common phenomenon in DNA and protein structure. Methyl transferase uses a reactive methyl group that is bound to sulfur in S-adenosyl methionine, which acts as a methyl donor. Methylation normally occurs on cytosine residue in DNA sequence. In protein, methylation occurs on nitrogen atom either on N-terminus or on the side chain of protein. DNA methylation regulates gene or silence gene without changing DNA sequences as a part of epigenetic regulation. So in bacterial system, methylation plays a major role in preventing their genome from degradation by restriction enzymes. It is a part of restriction modification enzyme in bacteria. Restriction enzymes cleave within the recognition sequence if the DNA is unmethylated. On methylation by methylase, the restriction enzymes are habituated by cleaving within the restriction site. Significance: inhibit binding transcriptional machinery proteins to DNA, thereby blocking gene expression. Recruit specific repressors at site that then switch off nearby genes, gene silencing, often by recruiting histone modifying enzymes. Allow discrimination between parent and daughter strands during DNA repair. Maintenance methylases protect DNA from action of endonuclease, which are part of restriction modification system. Then phosphatase. Phosphatase catalyzes the cleavage of phosphate group from substrate by using a water molecule. Hydrolytic cleavage. This reaction is not reversible. This shows totally opposite activity from enzyme like kinase and phosphorylase, phosphatase, that add a phosphate group to their substrate. This is phosphatase, removing phosphate. On the basis of their activity, there are two types of phosphatase, acid phosphatase and alkaline phosphatase. Acid phosphatase, it shows its optimal activity at pH between 3 and 6. A lysosomal enzyme that hydrolyzes organic phosphates, liberating one or more phosphate groups. They are found in prostatic epithelial cells, erythrocyte, prostatic tissue, spleen, kidney, etc. Alkaline phosphatase. It is a homodimeric enzyme which catalyzes reactions like hydrolysis and transphosphorylation of phosphate monoester. They show their optimal activity at pH of about 10. During post-translational modification, alkaline phosphatase is modified by N-glycosylation. It undergoes a modification through which uptake of 2Zn plus 2 ion and 1 Mg plus 2 ion occurs, which is important in forming active site of that enzyme. Alkaline phosphatases are isolated from various sources like microorganisms, tissue of different organs, connective tissue of invertebrate and vertebrate, and human body. Then polynucleotide kinase. It is a homotetramer with phosphatase activity at 3' end and kinase activity at 5' end with a tunnel-like active site. Polynucleotide kinase PNK catalyze the transfer of a phosphate group from Y position of ATP to the 5' end of the DNA or RNA and nucleotide monophosphate. PNK can convert 3' PO4 5' OH ends into 3' PO4 5' PO4 ends, which blocks further ligation by ligase enzyme. PNK is used to label the ends of DNA or RNA with radioactive phosphate group. T4 polynucleotide kinase is the most widely used PNK in molecular cloning experiments, which was isolated from T4 bacteriophage infected E. coli. Then DNA glycosylases. These are the enzymes that generally recognize damaged base nucleotides present in DNA and remove them. DNA glycosylases. They hydrolyze the glycosidic bond between sugar and nitrogen base, part of DNA repair system, producing abasic sites in the DNA. They are one of the key components of general DNA repair system. Then topoisomerase. These are enzymes that change supercoiling topology of DNA molecule. They generally remove supercoils, revealing the torsional stress present on the overwound DNA during replication. They transiently break the DNA backbone, thereby allowing the DNA supercoils to relax. Topoisomerase are classified according to mechanism which they use for changing DNA topology, type 1A and 1B. They break one of the two strands of DNA, rotate the end of the broken strand around the intact strand, and then seal the ends. The reaction does not require ATP. Type 2. They cleave both strands during reaction, rotate both ends by 360 degrees, and then reconnect the respective ends. The reaction is powered by ATP hydrolysis, DNA gyrase of E. coli. So once again have a quick recap or recollection of this enzyme. DNA modifying enzymes and significance. DNA modification refers to changes or alterations occurring in DNA molecule during replication, transcription, repair, genetic recombination, epigenetic regulations. DNA modifying enzymes, composition modifiers, topology modifiers, nucleases, exonuclease and endonuclease, S1 nuclease, restriction endonuclease, then deoxyribonuclease, deoxyribonuclease 1, deoxyribonuclease 2, ribonuclease A, ribonuclease H, DNA polymerase 1, DNA polymerase 2, DNA polymerase 3, Taq DNA polymerase, then ligases, methyltransferase, methylase, phosphatases, polynucleotide kinase, DNA glycosylase and topoisomerase, type 1A and 1B, type 2. Next topic, DNA repair mechanism. Since most mutations are deleterious, DNA repair systems are vital to the survival of all organisms. If DNA repair systems did not exist, spontaneous and environmentally induced mutations would be so prevalent that few species, if any, would survive. In humans, an individual who is defective in only a single DNA repair system may manifest various diseases and symptoms, including a higher risk of skin cancer. This increased risk is due to the inability to repair UV-induced mutations. Living cells contain several DNA repair systems that can fix different types of DNA alterations. Each repair system is composed of one or more proteins that play specific roles in the repair mechanism. In most cases, DNA repair is a multi-step process. First, one or more proteins in the DNA repair system detect an irregularity in DNA structure. Next, the abnormality is removed by the action of DNA repair enzymes. Finally, normal DNA is synthesized via DNA replication enzyme. So, UV light, pyrimidine dimer, DNA phosphorylase, visible light, like this DNA repair happening. So, types of DNA repair mechanisms. Direct reversal repair. Direct reversal repair is when an enzyme recognizes an incorrect alteration in DNA structure and directly inverts it back to a correct structure. An example of repair by simple reversal of damage is photoreactivation. Photoreactivation directly reverses the formation of pyrimidine dimer that result from ultraviolet irradiation. In photoreactivation, the enzyme DNA photolyase captures energy from light and uses it to break the covalent bonds linking adjacent pyrimidines. Another example of direct reversal is the removal of methyl group from the methylated base O6 methyl guanine. In this case, a methyl transferase removes the methyl group from the guanine residue by transferring it to one of its own cysteine residues. It is costly to the cell because the methyl transferase is not catalytic. Having once accepted a methyl group, it cannot be used again. Then mismatch repair. The structure of the DNA double helix obeys the AT to GC rule of base pairing. During the normal course of DNA replication, however, an incorrect nucleotide may be added to the growing strand by mistake. The structure of DNA double helix obeys the ATGC rule of base pairing. During normal course of DNA replication, however, the incorrect nucleotide may be added to the growing strand by mistake. Learning about mismatch repair. This produces a mismatch between a nucleotide in the parental and a newly made strand. Various DNA repair mechanisms can recognize and remove this mismatch. DNA polymerase has a 3' to 5' proofreading ability that can detect mismatches and remove them. However, if this proofreading ability fails, cells contain additional DNA repair systems that can detect base mismatches and fix them. An interesting DNA repair system that exists in all species is the mismatch repair system. The molecular mechanism of mismatch repair has been studied extensively in E. coli. 3 proteins designated MUT-S, MUT-L, MUT-H detect the mismatch and direct the removal of the mismatched base from newly made strand. These proteins are named MUT because of their absence leads to a much higher mutation rate than occurs in normal strains of E. coli. The role of MUT-S is to locate mismatches. Once a mismatch is detected, MUT-S forms a complex with MUT-L. MUT-L acts as a linker that binds to MUT-H by a looping mechanism. This stimulates MUT-H, which is bound to a hemimethylated site, to make a cut in the newly made non-methylated DNA strand. After the strand is cut, MUT-U, which functions as helicase, separates the strands, and an exonuclease then digests the non-methylated DNA strand in the direction of the mismatch and proceeds beyond the mismatch site. This leaves a gap in the daughter strand that is required by DNA polymerase and DNA ligase. The net result is that the mismatch has been corrected by removing the incorrect region in the daughter strand and then resynthesizing the correct sequence using the parental DNA as a template. Excision repair. There are two types of excision repair mechanisms. First, base excision repair. Base excision repair, or BER, involves the function of a category of enzyme known as DNA N-glycosylase. This type of enzyme can recognize an abnormal base and cleave the bond between it and the sugar in the DNA backbone, creating an apurinic or apyrimidinic site. Living organisms produce multiple types of DNA in glycosylase, each recognizing particular types of abnormal base structures. Base excision repair involves the function of Base excision repair involves the function of a category of enzymes known as DNA N glycosylases. This type of enzyme can recognize an abnormal base and cleave the bond between it and the sugar in the DNA backbone, creating an apurinic or apyrimidinic site. Living organisms produce multiple types of DNA N glycosylase, each recognizing particular types of abnormal base structures. Depending on the DNA N glycosylase, this repair system can eliminate abnormal bases such as uracil 3, methyl adenine, 7 methyl guanine, and pyrimidine dimers. Base excision repair is particularly important for the repair of oxidative DNA damage. Here, the DNA contains uracil in its sequence. We are talking about DNA repair via the N glycosylase. DNA contains uracil in its sequence. This could have happened spontaneously or by the addition or action of chemical mutagen. N-glycosylase recognizes a uracil within the DNA and cleaves or nicks the bond between the sugar and base. These release the uracil base and cleaves behind an apyrimidinic site. This abnormality is organized by a second enzyme AP endonuclease, which makes a cut on the 5' side. Following this cut by AP endonuclease, one of three things can happen. In some species such as E. coli, DNA polymerase 1, which has a 5' to 3' exonuclease activity, removes DNA segment containing the abnormal region and at the same time replaces it with normal nucleotides. This process is called Nick translation, although DNA replication, not mRNA translation, actually occurs. Alternatively, in eukaryotic species such as humans, the DNA is repaired in two possible ways. DNA polymerase beta has the enzymatic ability to remove a site which is missing a base and then insert a nucleotide with the correct base in its place. Another possibility is that DNA polymerase sigma or eta can synthesize a short segment of DNA which generates a flap. This flap is then removed by flap endonuclease. Nucleotide excision repair. An important general process for DNA repair is the nucleotide excision repair system. This type of system can repair many different types of DNA damage, including thymine dimers, chemically modified bases, missing bases, and certain types of crosslinks. In NER, several nucleotides in the damaged strand are removed from the DNA, and the intact strand is used as a template for synthesis of normal complementary strand. NER is found in all eukaryotes and prokaryotes, although its molecular mechanism is better understood in prokaryotic species. In E. coli, the NER system acquires 4 key proteins designated UVRA, UVRB, UVRC, and UVRD, plus the help of DNA polymerase and DNA ligase. UVRA, B, and C, D recognize and remove short segment of damaged DNA strand. They are named UVR because they are involved in ultraviolet light repair of pyrimidine dimers, although the UVRA-D proteins are also important in repairing chemically damaged DNA. A protein complex consisting of 2 UVRA molecules and 1 UVRB molecule tracks along the DNA in search of damaged DNA. Such DNA has a distorted double helix which is sensed by the UVRA-UVRB system. When a damaged segment is identified, the 2 UVRA proteins are released and UVRC binds to the site. The UVRC protein makes cuts in the damaged strand on both sides of damaged site. Typically, the damaged strand is cut 8 nucleotides from the 5' end of damaged and 4 to 5 nucleotides away from the 3' end. After this process, UVRD, which is a helicase, recognizes the recognition and separates the two strands of DNA. This releases a short DNA segment that contains the damaged region, and UVRB and UVRC are also released. Following the excision of the damaged DNA, DNA polymerase fills in the gap using the undamaged strand as a template. Finally, DNA ligase makes the final covalent connection between the newly made DNA and original DNA strand. Then recombination, one of the many types of DNA damage that can occur within living cells. The breakage of chromosomes called DNA double strand break or DSB is perhaps the most dangerous. DSB can be caused by ionizing radiation, X-rays or gamma rays, chemical mutagens, and certain drugs used for chemotherapy. In addition, reactive oxygen species that are the byproducts of aerobic metabolism can cause double strand breaks. The two main types of recombination repair are homologous recombination repair, HRR. Homologous recombination repair, also called homology directed repair, occurs when homologous DNA strands, usually from a sister chromatid, are used to repair a DSP in the other sister chromatid. First, the DSP is processed by the short digestion of DNA strands at the break site. This processing event is followed by the exchange of DNA strands between broken and unbroken sister chromatids. The unbroken strands are then used as templates to synthesize DNA in the region where the break occurred. Finally, the crisscrossed strands are resolved, which means they are broken and then rejoined in a way that produces separate chromatids. Then non-homologous end joining or NHEI, EJ. During non-homologous end joining, the two broken ends of DNA are simply pieced back together. This mechanism requires the participation of several proteins that play key roles in the process. First, the DSP is recognized by end-binding proteins. These proteins then recognize additional proteins that form a cross bridge that prevents the two ends from drifting apart. Next, additional proteins are recruited to the region that may precede the ends of the broken chromosome by digesting particular DNA strands. This processing may result in the deletion of a small amount of genetic material from the region. Finally, any gaps are filled in via DNA polymerase and the DNA ends are ligated together. Next, non-homologous end joining or NHEJ. During non-homologous end joining, the two broken ends of DNA are simply pieced back together. This mechanism requires the participation of several proteins that play key roles in the process. First, the DSP is recognized by end-binding proteins. These proteins then recognize additional proteins that form a cross bridge that prevents the two ends from drifting apart. Next, additional proteins are recruited to the region that may process the ends of the broken chromosome by digesting particular DNA strands. This processing may result in the deletion of small amount of genetic material from the region. Finally, any gap are filled in via DNA polymerase and the DNA ends are ligated together.